Good morning. Today we can discuss about the production of human insulin. Insulin, a peptide hormone which is produced by the beta cells of islets of Langerhans, which help to catabolize blood glucose. That means to decrease the blood glucose level. In conventional method, there are the insulin is extracted from the pancreas of bovine and pig pancreas, then directly injected into the diabetic patients. So, when the deficiency occurs, that lead to diabetes. So, due to the advancement of science and technology, there are due to the advancement of science and technology, insulin production is become more easier. So, we can see how the insulin is produced for mankind. First, isolate the human cells and grow in tissue culture. That means, in mass culture we can get in the tissue culture lab, then isolate the DNA from the human cells. That means, from this the gene for insulin is present. So, genes, all of you know that the small fragment of DNA which carries the information from one generation to next generation. So, when we isolate the DNA from these cells, that is the gene which produce for insulin. In the second step, when we isolate the DNA from this isolate the DNA from this, that should be cut into pieces by using an enzyme called restriction endonuclease. Why this enzyme is called restriction endonuclease means, this will cut at specific site of the DNA. So, that means the fragment is the insulin producing gene. So, when we cut into simultaneously, we should isolate a plasmid, that means a circular double stranded DNA, extra chromosomal DNA present in bacteria. So, these plasmids can also undergo replication along with the bacterial genome. So, when we take this plasmid, actually this is a gene carrier, or this will carry the desired DNA fragment. So, this gene carrier can be inserted into the plasmid DNA. Before that, we should cut the plasmid DNA by using the restriction endonuclease enzymes. See, for this, the figure number 5 indicate that the restriction endonuclease, when we use the same one, it will help to cut the plasmid DNA which will match to the desired DNA to anneal with that region. Then in the sixth figure indicate that we can join after cutting the plasmid DNA, then should be joined the desired DNA fragment or the insulin gene should be joined with the plasmid DNA. So, by using an enzyme called DNA ligase. So, then we will get the recombinant plasmid. Then this recombinant plasmid is again inoculated into the bacterial cell, that we can see in the figure 7. So, here in this, actually what is happening is that this recombinant DNA will enter into the bacterial cell. So, that here it will undergo, along with this bacterial genome, it will also undergo a replication. So, in the next figure, that is, this is a replication of how this plasmid DNA undergo replication in this. When this plasmid DNA is inserted into bacterial cell, so along with the bacterial DNA or the genome, this will also undergo multiplication. So, here we will get trillions of cells or many number of bacterial cells with the same or insulin gene can be get. So, these can be grown in mass by using a fermenter, and then this product, this gene will undergo translation. First, it will undergo transcription. All of you know that transcription and translation are the two processes. Here, this will undergo translation, and the product is the insulin, protein insulin. So, the commercially produced or by using the genetic engineering or gene technology, when we produce the insulin, it is called as Humulin. This was first produced by Eli Lilly and company and Gentech Corporation in 1980. So, to conclude or to sum up this is the summary of how we can produce the insulin. Take the desired gene of interest, then join with the plasmid, then to the recombinant DNA will get, then we allow or enter this recombinant plasmid into this, that will undergo multiplication. So, from this we will get multiple copies of the same gene. So, this is called as gene technology, or this is how we can produce human insulin. Actually, this is a boon for the diabetic patients. Thank you.